Okay, I'm sure I've talked about glyphosate in this particular presentation, or maybe not, but I've talked about it enough, you should know about it. Glyphosate is the thing that's in Roundup that kills all the weeds. But it also goes into the plants because the plants that they make are modified so they won't die from this stuff. So they eat the glyphosate and they're just happy with it. But you eat the plants and you eat the glyphosate, and look at this, glycerol. What was that? With few exceptions, cellular membranes, including plasma membranes and internal membranes, are made of glycerophospholipids molecules. Glycerol, a phosphate group and two fatty acid chains. It's the backbone of these membranes. Stephanie Seneff is the one that should be put in charge of this. She knows this stuff. And we need to get somebody that really cares about the chemistry and about what it can do downstream. Because upstream, where this is done, yeah, it's fabulous. Yeah, we don't have to weed. We just let those things grow. Man, they'll just grow out there all day long. And they do. And we have tremendous harvests. Nobody has to work. I think we should do it the other way around. Let them work. We're still going to get good harvests. And then we may all feel healthier. Okay, this is about this gut enzyme, IAP, is found to prevent aging in animal models. Now, IAP, again, enzymes and viruses are specific to species. Now, is that an enzyme? Is it a virus? What is this IAP? But they looked into it. I mean, we must have it too, I would imagine. Why would they look into it just to figure out about mice? So it says it's now accepted that gut barrier dysfunction and gut-derived chronic inflammation play a role in human aging. But how the process is regulated is still largely a mystery. So they're looking into it, and they're studying mice and fruit flies. Now, I looked into this study of the mice. Absolutely unbelievable. They took a control group, and then they took a group with some kind of oil, olive oil, I think it was. Yes, they took just water, olive oil, and then they took one with olive oil and the this IAP. And after so many years or so many months, every one of the water ones died, and almost all of the non-water ones that had both. And it was also, the oil ones did a little better, and the oil and the alcohol, this IAP, none of them died. They lived almost twice as long, they said. Absolutely amazing. It says, in other words, the enzyme maintained the composition of the gut bacteria, controlled the low-grade chronic inflammation that can happen with aging. There's so much to look into this. I'm not saying I have all the answers. I'm not saying I have any answers. I'm saying I have a hell of a lot of questions. I got some answers. I'm not going to give them. I mean, I'm not going to say these are answers to your situation. Whatever I talk about on my own. But I just talk. So if you want to look into it, well, you look into it. But I'm never going to tell anybody to do this or do that. Because I got to be honest with you, I have been wrong so many times that it just could be just another day. But I look in pretty close, and I think I got as many answers as anybody else is going to have on the same topic with the same information. There's a lot to think about, and I have a lot of back stuff to bring into this, because I understand these rocks. I understand the biology now, the chemistry, all of these things, and I look deeply into the texts, and I see nothing that doesn't work. I see nothing that doesn't. It's all based in fact. And then you go into the health. That's just a bit of it. But I can tell you this right now. In my world, God programmed all of the molecules to work with all of the bacteria and viruses and everything else. Some people are going to get sick, absolutely, but I think they are trying to work outside of God's natural prescriptions, which is healthy food, and none of this, glyphosate is that really concerns me. And Stephanie Seneff too, and then this other guy Morley something. I think maybe his last name is Morley, but it may even be in this video that they were talking about this. Very distressing, the things that they were saying about glyphosate, and it's ubiquitous. It's everywhere. You can't stop it. It's because it's capitalistically advantageous. And if you can make more money with something, you could justify a hell of a lot. That's what I have found. By the way, you should see this. Dr. Stephanie Seneff and Morley Robbins, this guy here, together on viruses, vaccines, glyphosate, and copper. She is fabulous. This guy was pretty impressive, too. And this guy's a doctor, Jason Dean. Very good. And of course, I go up there and run my mouth a couple times. And I put my own finger up. Nobody says anything. I don't know. Sometimes I think I'm just all by myself. I don't know if people even seeing the stuff I put up. I don't know. Anyway, you should follow her. She's a hero. Okay, my friends, Roger once again. Oh, Kiss Me Y'all sent me this link to look at this report by Dr. Rashid Buttar. And this is about the coronavirus thing. Now, he's got 2.2 million subscribers. The guy's got to be doing something right. He's a doctor. He's got 621,000 hits since March 27th on this, and it discusses the origin of coronavirus. And I found it pretty interesting. Now, I'm going to start here at 147. This is from, like I said, Video Advice, 2.24 million subscribers. Now, what we're going to do here is we're going to see what is this thing made out of and where did it come from, what's the ramifications of the damn thing. And they say it's been labeled as a bioweapon by some people, and of course the media says that's conspiracy theory, but let's take a look. I'm going to let him speak, and I will talk about this after he's done.
Conspiracy theory, but let's look at the actual configuration of what this virus, what COVID-19 is. So the first thing is I want to say that this wasn't because people were eating bat soup, okay, or transferred from snakes or sloths or whatever they said. The virus is not a naturally occurring phenomenon. This is very important, and I'm going to show you evidence of that. Now, this virus was actually created at the University of North Carolina in Chapel Hill, and they published the highlights of the creation in November 2015. The list of scholars associated with this Frankenstein of a virus, you'll see, named Zheng Li Shi, who just happens to represent the key laboratory of special pathogens and biosafety, Wuhan Institute of Virology, Chinese Academy of Sciences, Wuhan, China. Now, Dr. Shi, she's a key figure here. In 2014, Dr. Shi was the recipient of a number of U.S. government grants, as well as grants from the National Basic Research Program of China, the Chinese Academy of Science, the National Natural Science Foundation of China, and from the Strategic Priority Research Program of Chinese Academy of Sciences to assist in funding research into coronaviruses. Now, this is, remember, 2015. The U.S. government paid Dr. Shi and the Chinese government to fund research into coronaviruses. This was published in November 12, 2015, and this is actually the full article, the full research paper that was done. But what I want to do is I want to bring your attention to this March 2020 note that has been put there by the editors. And I want to read that first, and I'm going to go through the study, and then I want you to decide what this really means. So the editor's note before we start says, "We are aware that this story is being used as a basis of unverified theories that the novel coronavirus causing COVID-19 was engineered. There is no evidence that this is true. Scientists believe that an animal is the most likely source of the coronavirus." Now, it's obviously whoever wrote this, from the editor's note perspective, was either an idiot or doesn't understand the English language, but certainly wasn't a scientist, because I'm going to now show you what the study says. And remember, this is a warning now that they're putting here to dissuade people, to prevent them from actually looking at the science, to tell you that, look, the sky is not blue. Yeah, it looks blue to you.
I want to just make a point right at this point here, listening to this. This is science's immediate response to anything that disrupts somebody's academic success. Because once they are academically and accepted in this realm, they become the authority. Now, so you go to them and say, guess what, buddy? You got this all wrong. Well, you might as well walk out the door by yourself, because that's exactly what's going to happen. They will not listen. And this is across the board. You're hearing this from everybody. Now, it's just not me. I mean, I'm a complainer, absolutely. But I got rights to complain. And everybody else is complaining now, too, because they finally got the balls to stand up and say something about it.
To you, but it's not really blue. In November of 2015, an experiment that created a hybrid version of a bat coronavirus, one related to the virus that caused SARS, severe acute respiratory syndrome, as we discussed before, has triggered renewed debate over whether engineering lab variants of viruses with possible pandemic potential is worth the risk. This is the lead component, the first paragraph of this study that was published in Nature, probably.
Let's go back to see exactly what that said again, because that is so, this is unbelievable, absolutely incredible. What does it say? An experiment to create a hybrid version of a bat coronavirus. All right, so they had an experiment. It created this crazy version of a bat coronavirus, one related to the virus that causes SARS. You're not supposed to get through the different species. It's not supposed to happen, which is severe acute respiratory syndrome. It triggered renewed debate over whether engineering lab variants, which are changes to viruses, with possible pandemic potential is worth the risk. Is it worth the risk? Yeah, what the hell, let's give it, who cares?
Is worth the risk. This is the lead component, the first paragraph of this study that was published in Nature, probably one of the most respected journals in the world. So although almost all coronaviruses isolated from bats have not been able to bind to the key human receptor, which is this SHC014, which is the specific coronavirus we're talking about, was able to get into the...
Let me just explain you something. These are attachment sites. You see all these little bumps and knobs and things? They have different electrical potentials that attach to different molecules. That's the whole idea of viruses. And you have to have... That's why they're not supposed to go back and forth through species. Humans are supposed to have some kind of viruses, and rats are supposed to have other ones, and something else. Well, we engineered all kinds of things. We've changed all this stuff, and then we say, well, guess what? This doesn't cause any problem. Well, you don't even know what the hell you're testing it on anymore. The lab rats and lab mice are so screwed up now, and nobody will pay any attention to it whatsoever. I watched... I forget what the guy's name is. Him and his brother were talking about it the other day on a video. Absolutely, and they're doctors. And they say nobody cares. They don't care what they're using. They're just doing it. Just like putting one foot in front of the other. Don't care where you're going. All right, here's what I was just complaining about. Broken mice. The Epstein story where he had this sex ring and all this stuff. All these other problems is the media broken because they don't cover any of this stuff. They don't put forward the things that are of importance. And they say, oh, nobody's interested in that. And this, you should watch this. It's an hour long, so it's a little bit of a time consumer, but it's... It'll show you where we really are. These are doctors. This guy and his brother, they're both... They're Brett and Eric Weinstein. And we'll just listen to what he has to say. This is the interviewer. This is the guy that is going to fill you in on a reality.
Happened since, whether there's been any... Because one of the other questions that it brings up is what stories are being covered by and registering in sort of the mainstream, and which ones are sort of disappearing without trace, and why they might be disappearing without trace.
Good guy.
And what Eric's called the DISC, the Distributed Information Suppression Complex.
Yeah.
Maybe we could talk...
Distributed Information System Complex. Well, they have it, I would say, Distributed Information System crushed. in my case, or in these guys' case too. Anybody, it doesn't matter who you are. I don't have any credentials, so they're not listening to me just because I'm nobody. But these guys, they should be listened to. I don't care.
Talk about like what effect it's had, and just sort of without summarizing the whole story, why this is significant.
So the important piece of the story from the perspective of your audience and the rest of us is that when I was a graduate student, I was working on a question that involved cancer, senescence, and evolution. And I stumbled on a result in the literature that I had to grapple with that didn't fit with my hypothesis or anything else. And as I wrestled with it, the conclusion was that all mice, or in some cases all rodents, were thought to have long telomeres. Telomeres are repetitive DNA sequences at the ends of chromosomes, and at the point that I was doing this work in the late 90s, they were understood to shorten every time a cell divided. So when a cell divided its DNA to copy it, a little bit of the telomere was lost each time, and there comes a point at which a cell has a small enough set of repeats in its telomere that it no longer divides. I was working on the hypothesis that these shortening events were actually a protection against tumors and cancer, and that the downside of that system of cancer protection was that our tissues couldn't repair themselves indefinitely, and we grew old and feeble with age and ultimately die. The problem was that with the conclusion that all mice have ultra-long telomeres, this could not be made sensible, because mice have short lives, and if they have telomeres that are 10 times as long as people, they should have the ability to replace their tissues, and they should be very long-lived. So the podcast recounts the story of my increasing suspicion that there was something wrong with the conclusion that all mice have long telomeres, and my contacting a researcher, Carol Greider.
Now, first of all, just remember, the mice that he's working with are lab-bred mice. They're specifically bred for these experiments.
Who then tested that hypothesis with her graduate student at the time, Mike Heeman, and they discovered that in fact mice, that is to say wild mice, don't have long telomeres, and that it's only lab mice that do have long telomeres, as I had predicted. The most important outgrowth of that conclusion is that there's a problem in our drug safety system, because our drug safety system utilizes laboratory rodents, especially mice, to test the safety of drugs before they're ever tested on humans. Our mice, which have been altered by the environment and the laboratory breeding colonies from which laboratories get them, our mice have been altered by evolutionary forces in those colonies so that they have effectively an infinite capacity to replace damaged tissue and an incredibly extreme vulnerability to cancer. In other words, they eventually get cancer and their tissues do not grow old, right? Because their telomeres are so long.
All right, so what do I take away from this? This goes a long ways here, but it concludes with his brother coming on and saying, I can't believe these people. But I'm going to show you that, because his outrage I loved. Now, what do I take away from this? He says, well, they got long telomeres, they can reproduce their tissues forever. But they get cancer. Well, I can tell you why they get cancer. They get cancer because their microbiome has been so altered that the enzymes that are created by the natural chemistry of bacteria that no longer is the norm in their guts, it's gone. So they can't produce the enzymes that break up the chemistry to create the products, and they also break up the chemistry to make it small enough to attach to the carbons and go up there and blow them out, carboxylate this and do all that. It is a symphony. And if you can't, everybody's got to sing the right time, the right notes perfectly. We just threw a bunch of people in and don't know what they're doing. They're not supposed to be in there. These other molecules and these other changes that we've made to these mice's, microbiomes, and now all of a sudden they got all these long telomeres, and then we use them to figure out whether we're going to be healthy. Well, let's just cut to his brother's outrage, because I like that. All right, this is pretty good. He's going to talk about Vioxx. Here's another disaster. He says, okay, oh yeah, Vioxx works perfect in these mice.
Damage in our bodies, which will be particularly visible when it affects the heart, because the heart has a low capacity for cellular repair, and when the heart fails, it's very noticeable. So the idea is that something like Vioxx, which turned out to be destructive of heart tissue, was released into the public because our mechanism for testing whether it would be safe in mice had failed us. There are quite a number of other drugs that have shown the same pattern. We thought they were safe. They were then released. They turn out to do heart damage. They're pulled from the market, or warnings are appended to them. So the question is, have we made an error where something about our laboratory environments is creating mice?
Okay, I'm sorry, I got interrupted for a second. Here we go.
From anybody in the academy or
Nobody paid attention.
in the press, in the mainstream press.
There's some very strange thing where nobody seems to want to engage the issue that there was a first principles prediction coming from evolutionary theory that seems to have radical implications both for our understanding of death and for the way in which we use the mouse model to test all sorts of things. The mind reels. I can't fathom that, given the amount of interest in this story, and this video has just soared up the charts. It's now over 100,000 views. There must be 250,000 downloads, that came very quickly. Many people have heard it. And the interesting thing is how interested everyone is in the story, and how the interest is least in the place that you would expect it most. I would imagine if this was a wrong story, we would be hearing nothing but chortling from the molecular.
I have to chime in here again. I mean, I know I am interrupting like crazy, but I can't help it. I found this 100% across the board. Every single university I went to with my research. And my research is stunning. Stunning. It overthrows a ton of their research. And that's why they will not look at things exactly like this. They do not care a damn bit. Okay, well, I do say what I feel, and I'm telling you, I am feeling this to the very bottom of my soul. And I don't know what the hell is going on with the people that we trusted, but I do not trust them anymore at all.
Molecular biology community telling us how badly we've understood what in fact is going on. Or I would want to understand from the news media, tell me, why is this uninteresting to you? I should say that I've named these sorts of stories anti-interesting. An anti-interesting story isn't really uninteresting. The more riveting the anti-interesting story gets, the less interest there is from the official channels.
And why do you think that's the response from journalism? Let's start from
They're
molecular biology.
afraid.
Why do you think there's that response from molecular biology or from the scientific community?
Well, let's think about it. What if you had 20 years of studies that were built around a mouse model which was known to be broken? And that in fact you had people who were trying to say that the mouse model might be broken, and we need to have a discussion about this immediately. And the community had decided that it didn't want to introduce the idea that this might have far-reaching implications. Perhaps many results that we have based on the use of these mice are off. You could have 1,000 strains, but if they share a common breeding protocol, you might have a situation by which all of those strains are affected because they're all subject to the same etiology, that the breeding rotation is in fact an evolutionary environment.
All right, we don't have to go any further with this breeding and all this stuff. My point is, I don't care what it relates to. There is a pushback against anything that interrupts somebody else's expertise. And they say, well, I'm the expert in this field, and forget about it. Just get out of here, because I don't want you around here because I'm the expert and I've made all these determinations and the case is closed. Well, the cases are not closed. There's not one single case I can find that's closed. I can find a lot of them that have not even been opened, and I refuse to be opened, but I find none closed. Well, at this point, it's just turned into a very long discussion. And this is a leg from a hominid creature that is obviously engineered. It's nothing like us. This is the tendinous material. It's like a plate. And Tish Egerton has a ton of these. And some of them are eroded down to a point where you can see every detail within these springs. And they are torsion springs. These are called no-toes. I'm sure I showed one of mine, or probably by now. Because, they're just, this is a new species. Why doesn't anybody care about that? I do. Okay, as I said, my research on these soft tissue fossils is stunning, absolutely stunning, and there is so much to learn from it, and it's exactly treated like the rest that I am speaking of right now. Now, like I say, I'm sure I showed this before, but this is a no-toe, and that's where, the same as your foot, it's exactly, virtually identical. This is where the fibia comes down, and on certain lighting conditions you can see where the ball of the heel is in there, but this is right there. You see that little depression? That's where the bone straddles the back. And this is, it's what it is. And the toes are built into these. This is not like a shoe or anything. The toes are part of the feet, just like hers were. And there's a bazillion of them. And they're not just little like these. They come gigantic giants. I have dozens and dozens of pictures of no toes. And this is a good size one. And there's some of them are rounded like that, and some of them are squared off like this. And this one here obviously was to a point where the toes were starting to come out. Now the ones that Tish have, hold on. All right, here's some of the ones that Tish has. And inside, going like that, there seems to be two metal shafts going down here. Hers, these are eroded away, but they would have been closed right in, just like mine. You see that? That eroded out of there, but that was closed in, I can tell. But they all have toes in there, and they all have this little thing here. It was the same, and this is blood. This one, the toes came out. Like little sausages plugged in there. And that looks like a wing nut that held on. There's a big pad that goes on the bottom. My end, the pad is also missing. Well, not completely missing, but it's partially gone. They seem to be big. All the ones I found, they're big like a ball on the bottom, rounded off on the bottom, not an arch like we have. Well, maybe I could be wrong. They might have some arches, but I got a lot of them that show, well, some of them show arches, some of them don't. There's a lot of variation here. This is not something that can't be looked at.